Okay, guys, welcome back. Let's continue our discussion on some of the history of microbiology. Uh, Before the pioneering work of Pasteur, there was this man, Ignaz Semmelweis, who was an obstetrician, Hungarian obstetrician, working in Austria. And he was overseeing the maternity wards, some of the maternity wards in Austria. Now, just to give you a perspective on what it was like in the 1800s here at this time, Oliver Wendell Holmes published a piece on puerperal fever. This is a sepsis that is contracted during childbirth, and mothers would die from this disease. Now, the incidence of this disease was quite bad in the 1800s, and even today it does play a role in the deaths of women that do contract infections during the childbirth process. But let's look and see what Ignaz Semmelweis was dealing with. So here we have in the pink a clinic that he was in charge of. And you can see the number of patients the percent of patients that were dying from sepsis that was contracted during childbirth. And that incidence was quite low. And it kind of hovered around 1% here. And it was comparable to the deaths that would occur within a Dublin maternity hospital along those years within the 1700s and early 1800s. Now, what he experienced, though, in the going into the mid 1800s in his particular clinic in Austria was this sharp uptick in the deaths of women giving birth in his clinic. Now, childbed fever is just a common name for puerperal fever, and today, like I say, it still plays a big role, but this was alarming. And just by chance, Semmelweis, one of his physician friends, due to an accident he had with a scalpel that he had used to actually do an examination of a woman who had died from puerperal fever, he had an accident with a scalpel he had used to do that examination, and he died from that accident, from that incision. So he started thinking there might be a link between the examination of the dead in autopsies and these deaths of women in the maternity ward. And many of the physicians early in the day in his clinic would do autopsies. They wouldn't properly clean their hands and so on. And then they proceed in the second half of the day to do these procedures on women that were giving birth. And what they were doing was basically transferring whatever was associated with those autopsies to these women, and they were contracting the disease. So what he did was he instituted an onset of chlorine lime water hand wash, and that was instituted in 1847. You can see here, and their incidence of deaths from women who are giving birth went way back down, right to the 1% or so that you would have expected. Now today it's not nearly 1%. Today it's a small fraction of percent of women that actually die from sepsis from giving birth. But it is a significant percentage if you count just women that die from childbirth. Puerperal fever is still something that we need to worry about. Okay, so this was kind of starting to refute that miasma theory we were talking about before, the idea that there are miasmatic odors in the air, there's something in the air. Well, here, there was something on an object. not just something in the air only that could cause disease, but there's something on an object, whether it be someone's hands or a surgical device, that could actually transmit the disease. So now in England, this is Sir Lister, and Sir Lister actually began to employ some of these aseptic techniques, like those you would employ in the bio lab. The attempt not to introduce some kind of contamination in an experiment in the laboratory. He was applying these ideas to the surgical arena. Sir Lister would use carbolic and phenolic compounds and sprays to clean surgical equipment. And this greatly decreased the infections that we're seeing post-op, post-surgeries. Okay? And in fact, his name Lister is where Listerine comes from. Okay? This is Florence Nightingale. Okay? Most of you have probably heard of her, especially those people that are looking at going into the nursing field. Florence Nightingale was known for her school, where she was a founder of modern nursing, and she trained hundreds of nurses in sanitary techniques. So it's this idea, again, of using this aseptic technique in the nursing field as well, and she embraced that. So this is why she's also known in this particular arena. Now, as a result of Pasteur's work, Semmelweis, Lister, these individuals, they were starting to kind of refute this idea that there was just bad air, that it was supernatural. There was actually something living in the air or on objects that was causing disease. So we needed a nice methodical procedure, some way that anybody around the world could apply a procedure to identify the culprit, identify the disease-causing agent. And this is what Robert Koch, who you see here, put together, is a set of postulates, basic steps whereby we could approach a diseased individual, whether it be a diseased human or a diseased member of livestock or diseased plants when it comes to crops, and approach that in a methodical way. All right, so these were Koch's basic postulates. Okay, so we see here, number one, the suspected causative agent must be absent from all healthy individuals, healthy organisms, but present in all diseased organisms. All right, so you might approach a diseased animal like a mouse, okay, and then find that they are carrying a particular bacterium or virus, let's say, or other microorganism that is causing the disease. The healthy individuals don't carry this, but the diseased organism does. Okay, so you identify that there is a diseased animal. Number two, the causative agent must be isolated from the diseased organism and grown in pure culture. All right, we're looking at this in lab now, right? So if we have a mixed culture, any diseased animal is going to have hundreds and thousands of bacteria associated with them, on them and in them, okay? So what we need to do here is separate all of those other organisms that would be common with the healthy individuals that aren't carrying the disease, and isolate all of these organisms, right? So we might have, let's say there would be hundreds, but let's say we have 10, just to make it simple. So we would have isolated, maybe through streak plating, like we've talked about in lab, okay? We might use the streak plate technique to separate these organisms out of a mixed culture. And what we now want to do is identify which one of those 10 is causing the disease. So now we need some test subjects, right? So here, number 3, it says the cultured agent must cause the same disease when inoculated into a healthy, susceptible organism. Okay, so we need test subjects at this point. Healthy, susceptible test subjects. Susceptible meaning that they are able to come down with this disease. They are enough like the original animal we're studying that they would succumb or show signs and symptoms of the disease process caused by the one organism, the one microbe that's causing that disease. So let's say we had 10 isolates over here, right? 10 different color colonies, 10 different organisms, and then we take 10 mice and we inject into each of those 10 mice the 10 different microbes we've isolated. We would expect one of those 10 mice to pass as a result of one disease-causing agent. Then what we'd want to do, and this is the 4th step, the same causative agent, the same organism or microbe causing the disease, must then be re-isolated from the inoculated diseased organism. Okay, so what we want to do then is find again that disease-causing microbe from the dead animal. Okay? You're basically reconfirming the original organism that you found early on. Okay? So we're going to have an organism that dies from a particular disease-causing agent. Everybody else is healthy. Second step, we're going to isolate these different organisms in pure culture. Number 3, we're going to take each one of those isolates and inject healthy, susceptible test subjects. Okay, in this case, 10. And then finally, one out of 10 would be affected by this, and we'd re-isolate the disease-causing agent from that organism. We've identified our culprit, right? We've found that organism that causes the disease. This is the basic set of steps that Robert Koch used. He actually used those steps to identify anthrax, tuberculosis as the disease-causing organism with tuberculosis, and the organism that caused cholera. So he actually was integral to identifying those 3 different ones. But these could be used now with this set of steps by anyone. All right, here's what your book actually says here. It's very similar, maybe just a little bit different phrasing. A suspected pathogen, so this is the disease-causing agent in the other image, must be found in every case of the disease and not in the healthy individuals. Makes sense. The pathogen is going to be isolated and grown in pure culture. Okay, so we isolate these different organisms. Healthy test subject, okay. Susceptible is key here. I would actually add that in here, right? That's the one thing I don't like about this particular set is I want to make sure they're susceptible test subjects, able to come down with the disease. And they would be infected with suspected pathogen. And then one of those organisms would have the same signs and symptoms as the disease found earlier. Finally, we would re-isolate and match that organism to one pathogen that we isolated. Okay? So this is a basic set of methods for proving a specific microbe or pathogen causes a specific disease. Now, you can read about this in actually chapter 15 of your book, chapter 15, section 2. Just that section is all you really need to read there. It tells you about Koch's postulates. And what I'd like to do before we go too much further here is to just recognize that there are some limitations to Koch's postulates. These were a great first step. Now that people were recognizing that these small little guys, these little microbes, were the culprit that were causing disease, whether it be to crops, to livestock, or to the human population. This was a great first step, but there are some drawbacks, some faults with this set of steps. So let's think about what would be some of the limitations to Koch's postulates. Just thinking about what those steps are, okay? One problem is that it may be not possible to actually grow the particular pathogen in pure culture. We may not know exactly how this organism would need to be sustained in a laboratory. Many organisms are very preferential for the host that they cause the disease in, okay? A good example would be Neisseria gonorrhea, okay? Neisseria gonorrhea causes a sexually transmitted infection, and it was years before we could figure out how to grow that organism in the laboratory. Now we've overcome that, but we haven't overcome that with many pathogens, I'm sure. Another quick example I'll give you, my father had an ear infection, bad ear infection years ago, and he survived it, but it went into his brain. It went through the skull, the mastoid portion of the temporal bone. If you guys took A&P, it went through it. And they tried to culture it, and they could not. So right there, Koch's postulates out the door. You can't really proceed and do any kind of testing if you can't isolate the pathogen. Another problem is there may not exist a suitable experimental host, either for ethical reasons. For example, some organisms only cause disease in humans, like the HIV virus. You can't do any real meaningful research with the actual HIV virus unless you have humans to work with. And we obviously have some ethical issues there. Another thing that might cause you to say this is not a suitable experimental host, maybe you have a disease that causes certain signs and symptoms in humans, but then you apply that pathogen to another mammal, thinking it's close enough to, let's say, mice or rats. They may have very different signs or symptoms, or may not succumb to that disease-causing agent at all, if it's specific to humans. Another limitation. This set of postulates assumes the pathogen is only found in diseased and not healthy individuals. We know that there are carriers for bacteria. For example, people walk around with H. pylori in their stomachs. It's thought that 50% of people carry that organism around in the lining on the lining of their stomachs. But only certain individuals have gastritis, have ulcers, and things like that. Most people walk around with no signs or symptoms at all. So this is another fault, another assumption that isn't necessarily true when it comes to organisms that we carry around. Koch's postulates assumes that healthy test subjects are equally susceptible. So we all know that some people have better immune systems than others, right? This idea that the pathogen would be found on one diseased animal, that's Koch's postulate number one, right? The fact that it would be only found in one individual, well, we've just suggested that some individuals could be carrying it around, right? And they're still healthy. But there could be differences in immunity, again, also throughout that population. And they may show different signs and symptoms because of those differences in immunity. So there are a lot of complexities here that Koch's early postulates didn't address. Here's another one. The disease could result from more than one pathogen acting in unison. There are some diseases that occur because of two organisms behaving together, infecting together. So if you isolate, like we were talking in the earlier slide, if we have 10 isolates and you take them separately and introduce them into 10 separate test subjects, well, the two that were once together causing the disease in the initial diseased animal, well, they've been separated apart now, right? So you wouldn't necessarily see any signs or symptoms that are diseased animals as a result of that experiment. So again, go and check out these postulates. They're in chapter 15, and let me know if you're having any trouble understanding them, okay? All right, so I think this is probably a good place to kind of talk about how the historical milestones with Koch's postulates, Pasteur, Lister's work, all of these individuals really brought forward this idea that yes, they're out there, they're in our water, they're in the air, they're on objects. And people started looking around their environment, recognizing there are these things we can't see, living things we can't see. And so cartoons like this kind of suggest how people were thinking that, oh my God, if we look in the river Thames in England, we're going to find all of these nasty things. I mean, most major cities would actually, their sewage would go into these rivers, right? And so they started to find these kinds of things were caused by our own waste if we didn't take care of that waste in the proper way. And this gave rise to this idea that there's a germ theory, right? This idea that there are actual living microbes that can cause disease. And so we now would start actually looking for some kind of chemical that would be a treatment for this, right? A chemotherapy. Now we think, when I say the word chemotherapy, you probably think about cancers, but chemotherapy is nothing but a chemical that's a treatment, that's also a treatment, okay? So now that we recognize that there were these organisms, we wanted to find their weak point, right? We wanted to find out what would kill just those particular kinds of cells. And this is where we started to look for chemicals like antibiotics and identify these kinds of chemicals, like Fleming's discovery of penicillin here. Here, the penicillin or molecule is being produced by a penicillium fungus, and it kills any bacteria that are found nearby. And the idea that we can isolate and purify these chemicals and introduce them into maybe a human or livestock or something like that, this never would have come about if we didn't recognize that there were germs, were living things that would cause these diseases. Here we have some other pictures of penicillium. Penicillium growing on a petri dish, like you've seen in the lab section of this course. Here we see some bacteria that's growing, and then penicillium is secreting this chemical. And what we're doing with antibiotics is kind of stealing this idea, right? Using this chemical, this warfare between microorganisms, in this case a fungus and a bacterium, and we're using it to our advantage. So we can do studies to identify effective antibiotics on petri dishes. And we're going there, okay? We're going to eventually get to that point in lab. All right, so I think this is probably a good point to leave off, all right? We're done with the basic kind of historical perspective, and next time we're going to jump right into the morphology of these bacterial cells. We're going to focus on bacteria, and it'll kind of merge with some of the things we're seeing in lab. All right? See you guys.